So today's video is about the cleavage. But before discussing cleavage, that you all know that through fertilization, that is in the ampulla region, that is zygote is formed. Now, uh, you all know that from this zygote, that is embryo has to be formed. So therefore, and for the formation of embryo, you can see here that this zygote undergoes so here repeated number of unequal divisions, and you can see here that and due to this repeated number of unequal divisions, that is the number of cells is increasing. Here number of cells is 2, here 3, 4, 8 and 16. So here, and this is the main concept of cleavage. And also you can see here as this a fertilized ovum or the zygote, because that is, you know that for the zygote formation, that is ovum must be fertilized at the ampulla, that is at the ampulla, and therefore zygote formation done at ampulla. Now as the fertilized ovum that is passes through the uterine tube or the fallopian tube, in that situation that this a number of unequal divisions is occurring, and as a result that is a number of cells are present in the fertilized ovum is also increasing. And this is the main concept of cleavage. And also definition, and also that is little bit information that this A is the zygote, and this B is the 2 cell stage, and this C is the 3 cell stage, and this D is the 4 cell stage, and this E is the 8 cell stage, and this F is the morula. So I hope you can understand the definition more clearly. The process of repeated unequal divisions of zygote or the fertilized ovum that leads to increase in number of smaller cells or blastomeres. As you can see here, that is a number of cells goes on increasing, and this process occurs within the zona pellucida. But when you will study that is implantation, then you will understand the importance of this zona pellucida. And also, and that process is known as cleavage. So this definition of cleavage done. So here duration, that is for 6 days up to 7th day after fertilization. It is an implantation, it should be fertilization. So that is simple, that is a duration. But now that is, you should also here understand the, so here that is difference between cleavage and mitosis. So here that you can see that here that this is a cell and this is a cell. Now here you can see here that the first thing that I think that you can see that this cleavage causes unequal division and this mitosis causes equal division. So due to cleavage, you can see here that is large cell and a small cell form. That is, all the cells they are formed, they are not of similar size. But in mitosis, you can see here that equal division occurs and all the cells formed, they are of equal size. And this is the main difference that you can see, that is I have written, that is in cleavage. So here in cleavage unequal division of a cell occurs, but in mitosis equal division of a cell occurs. So therefore that is a duration of cleavage, and here difference between cleavage and mitosis clear. So now that is I am going to explain about the subdivisions of cleavage. So therefore that there are 3 or 4, that there are 4 subdivisions, not 3, that is earlier it was 3, but there are 4 subdivisions in cleavage. So first one is a stage of compaction, second morula, third blastocyst, and the fourth one is the hatching of blastocyst. Now, so first of all, that you should understand a stage of compaction. But before understanding the stage of compaction, that is, you should see here that in 2 cell stage, all cells they are of similar shape and size. Here also, that is from 2 cell stage to 8 cell stage, you can see here all cells are of similar in shape, size, and they can form same derivatives from 2 cell to 8 cell stage. Now in this diagram also you can see here basically all the cells are similar to one another and they can form same derivatives. So therefore that is I have written, that is up to 8 cell stage all the cells are of similar shape, size, and they can form same products products are simple that they can form same derivatives. So therefore these cells are called totipotent cells. But now, so here comes the concept of stage of compaction. So here this is a 8 cell stage at ovum and this is 16 cell stage. Now here that you can notice that in a 16 cell stage that there are two different types of cells are there. So here that is inner cell mass and here basically outer cell layer or trophoblast. Now that is a stage at which basically of cleavage, that is a differentiation, basically the differentiation occurs, and as a result of which basically that in 16 cell states two different cell types form, inner cell mass and trophoblast. And here that is the most important thing is that is inner cell mass and trophoblast, they will not form the same products or same derivatives, they will give different derivatives, they have different shape and size also. So therefore they are non-totipotent cells. And that stage of cleavage at which this occurs, it is due to a process or reaction that is known as a stage of compaction. Therefore, I have written that during cleavage process, a stage occurs at each cell stage, due to which the cells of 16 cell stage are divided into two types, inner cell mass and trophoblast, as I mentioned. And since inner cell mass forms different derivatives and outer cell layer or trophoblast forms different derivatives, so therefore that these cells are totipotent, sorry, non-totipotent cells, and they have also different shape and size. So therefore that is stage of compaction. So therefore basically that is stage of compaction is responsible for the formation of trophoblast and inner cell mass in 16 cell stage. So now you can see here that is at the 16 cell stage, you can see here that is the fertilized ovum is look like that is mulberry, so therefore it is known as morula. But that is not important, but that is the important part is that two different types of cells, you can, as you already know, two different types of cells are there. So here inner cell mass and trophoblast. Now, and also that is still not zona pellucida is present, because that is invention I told that this cleavage occurs within the zona pellucida. So therefore within the zona pellucida that this cleavage occurs. So therefore now that is, as I already mentioned, that is trophoblast and inner cell mass form different derivatives, that is trophoblast forms the embryo covering and this, so here inner cell mass and this inner cell mass forms the proper embryo, and these trophoblastic cells provide nutrition to the embryo. Yes, basically, and in reproductive physiology, that is when you will study the, so here cycle, ovarian cycle, or how that this a pregnancy occurs, then you will have to important, so remember this, and this is responsible for causing decidual reaction also, decidual reaction, and also that is important for nutrition to the embryo. That is in physiology you will have to, reproductive physiology you will have to study in details about this. So now the third stage is about a blastocyst. So now that is, so now you can see here that is morula is within the uterine tube, that is fallopian tube. But when this so here morula reaches the uterus, so here you can see here that is from this uterus that is fluid accumulates, that is uterine fluid. So this is the uterine fluid, so this is the uterine, so this is the uterine fluid accumulates in the, so here that is morula. As a result, that is, as you can see here, initially that is all the inner cell mass cells, they are scattered everywhere, but now that this inner cell mass cells that is started to that is to be attached or present, that is only one side of trophoblast or only one side of the fertilized ovum, not that is around the whole fertilized ovum. So therefore the due to accumulation of the fluid, the inner cell mass are completely attached to the one side of the fertilized ovum and to one side of the trophoblast. and also that is a long cavity is formed that is known as blastocell. And now that is morula with the presence of blastocell is known as blastocyst. Until now trophoblast, thick zona pellucida, inner cell mass are there, and all these portions I've written. So therefore that you can see here between 4th and 5th day when the morula reaches the uterus. So here that this is the uterus. When morula reaches the uterus, then this uterine fluid, so here that is accumulates in the morula. You can see here as a result the inner cell mass get attached to the trophoblast on one side. You can see in the diagram that this inner cell mass gets completely attached to the one side of trophoblast, and also a large cavity called blastocyst form. Now the morula called blastocyst. And for further information, that is, so here polar trophoblast and mural trophoblast. So here a trophoblast, so here that is trophoblast site where this inner cell mass attached is known as polar trophoblast, and the remaining is known as mural trophoblast. So here trophoblast where the inner cell mass attached is called polar trophoblast, and remaining trophoblast portion where inner cell mass not attached called the mural trophoblast. Now here hatching of blastocyst, you can see here that is now that this initially it is thick zona pellucida, but now it is become thin, and this thinning of zona pellucida that is starts at 4th day, and this completely disappears at 5th day of fertilization. And this disappearance of zona pellucida initiates implantation, and this disappearance of zona pellucida is known as hatching of blastocyst. Hatching of blastocyst, because we'll understand that now this blastocyst is completely, so here basically attached to the uterine, that is the endometrium or the wall of uterus. And this is the basic concept of implantation, but in the next part you will have to study this. So now I'm going to discuss our principal effects of cleavage. So friends, you can see here that in zygote, that is the size of cytoplasm is very large with respect to nucleus. So here basically that is nucleus cytoplasmic ratio is very low, but here that is, as you can see here, as the cleavage so here occurs or the unequal division continues, that is the size of cytoplasm, so here cytoplasm size goes on decreasing. So therefore that this n by c ratio that is starts to goes on increasing, or it is maintained. So this is the main effect. So here restoration or the maintenance of nuclear cytoplasmic ratio in the fertilized ova. So here restoration or here maintenance of nuclear cytoplasmic ratio in the fertilized ova. You can see here also basically through the cleavage process, that is the number of cells, so here that is in the fertilized ovum goes on increasing, and this is the second important effect basically to increase the number of cells. So here increase the number of cells. So therefore, so here principal effects of so here cleavage M, so here subdivisions of cleavage completed. So now, so when you will write in a university exam, or when you will do the revision, or that is when you will have to remember, that is always try to first write, that is, so here that is start from here, what is a definition of cleavage, and second, so here duration of cleavage, and third difference between a cleavage and mitosis, and third so here fourth is the subdivisions of cleavage, and the last is the so here principal effects of cleavage. And if you write in this way, you will get a good marks in your exam, and also it will become very easy for you to remember the concept of cleavage also. So remember in this so first definition of cleavage, a duration of cleavage, difference between cleavage and mitosis, subdivisions of cleavage, and principal effects of cleavage. If you remember, you can easily remember the concepts of cleavage, and also that it will help you a lot to increase or to get good marks in your examination. That's all about the cleavage. Thank you.